So here we're going to understand gel filtration, or also referred to as size exclusion chromatography. Now, this is a chromatography technique used to isolate molecules based off of their size. Now, essentially, what happens is that we have this column filled with an inert matrix, but specifically we have these beads. Now, these beads are porous, and we can manufacture these beads to have pores of specific sizes, so that when we add a mixture of molecules to the top of the column, the smaller molecules who can fit into the pores will travel through the pores in the bead from one bead to another, and overall they will have a larger distance to overcome and then elute out from the column. Whereas those larger molecules that cannot fit into those pores will have a more of a linear path to overcome, and they will elute out of the column first because they have that shorter distance to travel. Just to kind of get a picture in our heads, so essentially, let's say that this is our bead, this is the pore opening, and then our molecule will travel through the pore, enter another bead, and travel through the pore again. So overall, it's just a larger different distance for smaller molecules. Now here we only have two different molecules, and we can see that these small pink triangles are going to move through the pores of the beads, longer distance to travel, whereas these purple squares do not fit through the pores, so they elute first because they have that shorter distance. And then we'll have a test tube down here, or a beaker, collecting the first larger molecules, and then we will have the smaller ones come in after, eventually eluting from the column. Now, what's important to note is that here we only have a bead with a single pore size that fits the pink beads, but realistically we could have many different molecules with different sizes. In that case, we will have pores and beads of various sizes, and then based off of that, the largest molecule will elute first, the second largest will elute second, and then so forth and so forth, and then the smallest molecules will elute last because they have the largest distance to travel because they can go through the pores. And that is essentially gel filtration, or size exclusion chromatography. You utilize an inert matrix, most commonly agarose, polyacrylamide, or polystyrene, and you have these beads that are porous and smaller molecules. Based off of the size of these pores, small molecules will enter, travel longer distance to overcome compared to the larger molecules, and this is just a chromatography technique that is used to isolate molecules based on their size.